This conference will now be recorded.
Good evening, students. I'm Dr. Divya on behalf of TeenEinstein.
Good evening, ma'am.
So, good evening. In the fifth chapter, till now we were learning about different concepts of genetics, right? So, genetics, that is a branch of biology which deals with the principles of inheritance and that different inheritance patterns or that practices, how it is getting inherited. About that we are learning in this chapter, principles of inheritance and variation. So the progeny, which is resembling the parents, that is in morphological and physiological way, it will resemble the parents. So this feature, that mainly that character or that inheritance pattern that attracted many biologists, right? So Mendel was the first person who studied this phenomenon in a systematic way. So while studying that pattern or that inheritance pattern, which material he used? Pea plant, Pisum sativum, for the studies, for his studies. So in that plant was having several contrasting characters, and Mendel proposed after his studies, all the studies, 7 contrasting characters he selected, and he conducted several studies in that. And after all these studies, he proposed the principles of inheritance. And today, we are calling this as Mendel's law of inheritance. So he proposed that some factors are regulating the characters. Nowadays, we are calling these factors as genes. So these factors, they are regulating the characters, and these factors, they are found in pairs. Okay, these pairs are called as alleles. So the factors now we are calling as genes, or earlier during Mendel's period, they were calling it as factors. So he observed that these characters are found in pairs, and these pairs, they are called as alleles. So he observed all these expression of characters in an offspring, that is getting segregated in a particular pattern from that parent to offspring. So there is a pattern of inheritance. Okay. So he observed the expression of characters in the offspring, and that is in a very particular pattern. From the first generation, it is getting to the second generation. That means from the F1 to F2, and like that it is continuing in each generation. Right. So the recessive characters, when that recessive characters and dominant characters will express dominant characters, whenever the dominant allele is present, it will express. But when a recessive character will get a chance to get expressed, only when it is expressed in homozygous condition, in that condition only a recessive character will get expressed. Recessive get a chance only when there is no dominant allele, or the presence of dominant allele will not be there. And that recessive allele should be in homozygous condition. If it is in heterozygous, it won't express because there will be a presence of a dominant allele, right? So when it is in a homozygous condition, the character will get expressed, the recessive character. And the character never blend in heterozygous condition. Okay, in heterozygous condition, that character will never get a chance because there will be a presence of dominant allele. Okay, so the recessive character that was not expressed in heterozygous condition, and again it will get expressed when it is in a homozygous condition. Okay, hence the that character will segregate while formation of gametes. That particular gamete formation, the segregation of these characters during gamete formation, that he proposed as law of segregation. Okay, that part Mendel proposed as law of segregation. So from this, all the characters, they won't show true dominance. Okay, some characters show incomplete. We learned about incomplete dominance, then codominance, all these things we learned. Just I'm telling a summary of this because today's topic that is related to or that shows slight difference or this is a different concept from the earlier studies. That's why I'm explaining all these things. So not all the characters, all characters will never show true dominance, right? And some characters will show incomplete dominance. Incomplete means there won't be, it will be dominant only, but the dominance will be in the form of incomplete or it won't be in a complete form. And some shows codominance. Codominance in the sense all dominant alleles will get expressed at the same time. There will be simultaneous appearance of the dominant alleles. Okay. So when Mendel studied the inheritance of two characters together, he found that all these factors, they are getting assorted independently and different types of combinations he saw, different combinations of gametes. And with the help of a Punnett square, we are representing this and it is called as Punnett square. Reginald D. Punnett, he suggested this and the factors on chromosome. They are regulating the characters and these are called as genotypes. The factors, that combinations, genetic combinations which we are getting that are called as genotype and the physical appearance or the physical expressions which we got that is called as phenotype. That is the physical expression or the appearance, how that organism is getting appeared in a particular genetic combination. The genetic combination is called as genotype and physical appearance or the expression of the genotype that is called as phenotype, right? So after all these, later they conducted more and more studies and they located these genes, where it is present and how it is working and how it is getting segregated during meiosis or the gamete formation, all these things. So we are starting those topics today onwards. Okay. So principles of inheritance and variation. Today we are going to discuss about polygenic inheritance. First we will discuss about polygenic inheritance. So let's start. Polygenic inheritance, that is otherwise called as quantitative inheritance. Polygenic inheritance is otherwise called as quantitative inheritance. Excuse me 1 second. Sorry. So we said about polygenic inheritance. So it is otherwise called as quantitative inheritance. Polygenic inheritance is otherwise called as quantitative inheritance. So what is polygenic inheritance or quantitative inheritance? Do you have any idea? Because till now, according to that Mendel's law, we were reading all these things, we were studying all these things. So till now, Mendel studies all those were mainly explains about the traits which are having alternative forms, right? We were learning about one character. See, for example, we are studying about plant height. Plant height, right? Mendel took one set of character, plant height, and He told like there is a tall plant and crossing with the dwarf plant. So till now we were explaining the concept according to this Mendel's idea. Like plant height of we are selecting a particular character, and in that two different forms, two alternate forms we are explaining. Okay. So just like a flower, it is having either purple color or white color. Like that also, while selecting the characters, like flower color, alternate forms of that trait, white color and purple color. But there are so many traits around us which we don't have a distinct nature. Like this flower color or this plant height, we can't explain that. We have so many traits like that, which we can see around us only, like how it will be. It is not like these two forms. Okay, here we can distinctly tell like there is a tall plant and there is a dwarf plant. But so many other traits are there which we can't make into these alternative forms. Like there is no two alternative forms, exact forms. We can't tell like this character will come and next another dwarf will come, this tall will come. It's not like that. So we don't have a distinct nature for so many characters. For example, in human, we can study tall people or short people. In human being, is it possible to study tall people and short people? In the case of human? See, I'm saying about the difference between this Mendelian concept and this polygenic inheritance. Or what is quantitative inheritance? I'm trying to explain with the example. In human beings, how can we tell tall or dwarf? We can't tell like that because it depends, right? Like you can't see people with different range of, see, this person is tall, person A is tall, and person B is dwarf. But this will work out only in the case of plants. In plant, we can select tall plant, dwarf plant. But there is a, in the case of human, you can see an intermediate. And here also, one more, little bit higher, which is having little more height. And here you can see in this condition. So all these ranges will come in between, right? So you can see people with different ranges of height. In the case of human beings, ranges. There will be different ranges. In the case of human beings, height, if we are considering the character height, we can see different ranges of height. So such traits, they are generally controlled by two or more than two genes. Okay. Here, this type of traits, like there is a wide range of or variations are seen in the character or wide range. There is a range for each character that may be controlled by more than two genes. Okay, two genes or more than two genes. Okay, so it is called as polygenic. Poly means more than or multiple. Polygenic, more than two genes. It is controlled by multiple genes. So polygenic inheritance. That's why it is called as polygenic inheritance. Polygenic inheritance. So what is that? A character which is controlled by more than two genes. So at that, from that word itself, you can make out poly. Poly means many or multiple. Polygenic means it is controlled by more than two genes. That is called as polygenic inheritance. So such type of traits, they are called as polygenic traits. So the polygenic inheritance, the characters or the traits which are controlled by more than two genes or polygenes, they are called as polygenic traits. Okay, those traits are called as polygenic traits. Okay, so the inheritance of polygenic traits, it is called as polygenetic or polygenic inheritance or quantitative inheritance. So these traits, the inheritance of these traits, which is controlled by more than 2 genes, that is called as polygenic inheritance. Okay, inheritance of polygenic traits, that is called as polygenic inheritance or quantitative inheritance. Okay, quantitative inheritance or polygenic inheritance. What is that? Inheritance of a character or a trait which is controlled by more than 2 genes. That inheritance is called as polygenic inheritance, and that particular trait is called as polygenic trait. Got it? So this type of traits, mainly they are controlled by multiple genes. Okay, there comes the concept of multiple genes. Up to this, we learned about one gene controlling one character. Like that, we learned. We selected one character, we selected that particular character and crossed. So like that, that is the concept which Mendel developed, right? So here comes more than 2 genes that is controlling one character. Okay. So in quantitative inheritance, multiple independent genes, they have additive or similar effect on a single quantitative trait. Okay. Suppose here one quantitative or a polygenic trait. Examples I'll explain later. So now consider one polygenic trait. Okay. This polygenic trait, in that multiple genes like gene A, gene B, gene C, gene D, all these genes will be having a particular effect on this trait. While expressing this trait will get expressed. Expression of trait. Expression of this trait depends upon the work or the expression of these genes, all these genes. Okay. So there is a, for a single quantitative trait, this is a single trait, but it is getting expressed with the help of all these genes. Okay. So there is a additive or similar effect. See, this gene will be having one effect. This gene will be having second effect. This will be having third effect, and this will be having fourth effect. But all these are having equal importance. Equally important. All these are equally important. Okay. So all these genes are having their own expression, but all are equally important. But all these together will work, and the expression of a trait is taking place with the help of multiple genes. Okay. So a quantitative inheritance, multiple independent genes. All these genes are independent. They have, while single, they have their own expression. Okay. When they are working together and they are forming an expression for a particular trait, that concept, they have an additive effect. They will work together. They will add their own expression, and that altogether it will form a expression of a trait or a trait. That is called as polygenic trait or a quantitative inheritance. So in this inheritance of a character, this is depending upon cumulative action of many genes. Here I said, for the expression of this character X, suppose this is the character X or trait X. For the expression of this character X, we need additive effect or the cumulative effect of each gene. Gene A, gene B, gene C, and gene D. All these genes will express, and that expression together will be working for the expression of this trait X. Okay. So each gene produces small effect. Each and every gene, the A gene will produce one expression or one effect, then B, C, D, each and every gene is having their own role. But full trait will be expressed when and all these alleles are present together. Then only the full expression of a trait will take place. Okay. Here, suppose X trait, X expression when the complete expression will take place, the expression of A plus B plus C plus D. All these genes are working together means that will form their own expression or their own role they will handle, and they will work together and they will produce a expression of this trait X. Okay. So the full trait will be expressed when all these genes, all these dominant alleles are present, then only it will be expressing in a complete form. So the expression when it will take place, see here A is in dominant condition, B is in dominant condition, but C is in recessive condition. Here recessive allele is coming and D also dominant. Then also the complete expression won't be getting, right? There won't be a complete expression of this A, B, C, D. Then the proper expression, the final expression of the trait X we can't expect the complete expression of this trait X. Okay, when all these are coming in proper form, in dominant form, and in all these genes they are getting expressed, that will work together. The cumulative effect of all these gene expression will form an expression of a particular trait. Okay. So here, scientists Nielsen and East, they demonstrated the segregation and assortment of genes controlling quantitative traits. No doubt, the scientists whom and all demonstrated that Nielsen and East, they demonstrated the segregation and assortment of genes, okay, which are controlling these quantitative traits. So they demonstrated the segregation and assortment of genes which are controlling these quantitative traits. Okay, they conducted studies in the genes which are controlling the segregation and assortment of genes which are controlling the quantitative traits. So polygenic trait means, what is that polygenic trait? It is called as polygenic or quantitative inheritance. Okay, so what is that? With expression of a particular trait with the help of or as a cumulative effect of many gene expressions. Okay, is it clear? Like polygenic trait, this trait is getting inherited. That inheritance is called as polygenic inheritance. So what is a polygenic trait? With the help of multiple genes, they are getting a particular trait is getting expressed. If any one of that gene, among that multiple genes, any one gene is not expressed in proper form, we cannot expect the final expression of that particular trait, which is supposed to be the cumulative effect of several genes. Okay, so the expression, when it is all these genes are coming in dominant form, then only the final trait expression will be complete. Otherwise, it won't be in a proper result, or that won't give a proper result. Okay. Then, what are all the characters of this polygenic inheritance? We need to learn the polygenic inheritance, the characteristics. Polygene, that refers to a gene that exert a slight effect on a phenotype along with other genes. Okay. Polygene means, just now I explained, that is, with so many genes, each gene is having slight effect, and that together it will get expressed. Polygenes, multiple genes. That to a gene that exert a slight effect on a phenotype. Each gene will be having their own role, their own hand in the expression level. Okay. So effect of a single gene is too small, so it is difficult to detect. I said so many genes will be there and each gene is having their own role. So it's very simple role. That role will be, or the single gene, it is too small. That role is too small, or that effect is too small. But it is very difficult to detect. And these multiple genes produce an equal effect. All these genes which are present. See, I said gene A, B, C, D, like that. 4 genes are present. Then multiple genes, they will produce equal effect. Each and every gene will produce equal effect. And each allele has a cumulative or additive effect. Okay. And from the each gene, that alleles will be getting into next generation, right? So that each allele is having a cumulative effect. Allele from gene A, gene B, gene C, gene D. All these are working together, or they have an additive or cumulative effect for the final expression. So the polygenic inheritance that differs from multiple alleles, as in multiple alleles, 3 or more alleles are present in the same locus of which any 2 alleles are present in an organism. Got it? Polygenic inheritance and multiple alleles, both are different concepts. Okay. In multiple alleles, that genes, that alleles, the alleles of the genes. For example, we can select ABO blood group, right? ABO blood group that are different. This is having different alleles. I gene, different. This is the gene for blood grouping. Okay. This I gene is having different alleles. So that is from same gene and the same locus. But in the case of polygenic, here for alleles, it is having different form IA, IB, and small I, right? This is from same gene, same locus. But in the case of polygenic inheritance, what happens here? Polygenic, that genes itself different. They have multiple genes. Okay. So all these genes, they are not from same locus. Okay. So don't get confused. Polygenic inheritance and multiple alleles, both conditions are different. In multiple alleles, there are more alleles, and these alleles are present in same locus. Multiple alleles means same gene is having more than 2 alleles. Here you can see more than 2 alleles, presence of more number of alleles. That is called as multiple alleles. Okay, but it will be in same locus because it is the allele of same gene. And any 2 alleles present means that will show the expression. Okay. So here ABO blood group system that is controlled by 3 alleles, and any 2 alleles like IA and small I is present means that will go for A group, and IA and IB is present means that will form AB group. Like that, presence of any 2 alleles will give an expression. Okay. But in polygenic inheritance, another characteristic, that is, there is no epistasis involved. Epistasis, what is that? Masking effect. Masking of the expression of one allele. Okay, one allele that is from different locus. So there is no masking effect of one gene over the other in polygenic inheritance, because I said this is the cumulative expression, right? Each gene will produce its own effect, expression, or that each gene is taking place its own role. Okay. So there is nothing like one gene is not working, so the same expression that will give. No, that is the cumulative result of the expression of several genes. Okay. So there is no masking effect involved in this. There is no epistasis, no masking effect. And there is no linkage or dominance. Rather, there exist contributing and non-contributing alleles, which are known as active or null alleles respectively. Okay. So there is no linkage or dominance. There is no dominance. I said there is no epistasis, right? So there is no dominance or no masking effect. But there is a chance of non-contributing alleles. See, in one gene, like suppose A gene, in that 2 alleles will be there. A and B, both alleles, capital A, dominant allele and recessive allele will be there. In that, some allele may be contributing for that final expression and some alleles may not be. Okay. So which are contributing, they are called as active, and which are not contributing, they are called as null alleles. Note down these active alleles and null alleles, these and all important for NEET exam. So there is no linkage or no dominance in the case of polygenic inheritance, but there is contributing alleles and non-contributing alleles. Okay. Alleles may contribute or that may not contribute. The contributing alleles are called as active alleles and non-contributing. They are represented as null alleles. Okay. And the polygenic inheritance. Actually, the polygenic inheritance, this is a very deep topic. You have so many terms, new terms, and so many. There is a different concept. So just note down all the points. It will be very difficult for you because you will get confused. So polygenic inheritance, it is characterized by continuous variation of the phenotype of a trait. Okay. This is characterized by continuous variation. Why it is getting variation? Because if you are saying one gene, this gene is expressing one character, it is red color or something, some character it is getting expressed. There is only one gene is involved. But in the case of polygenic inheritance, the character is by continuous variation. It is characterized by continuous variation because multiple genes are involved and you can see different forms of that gene. That means different alleles, right? So there will be continuous variation in that phenotype of a trait. So the polygenic inheritance pattern is complex. So the inheritance pattern of the polygenic inheritance, that is complex. We can't say that. That's why we said there is a range. In the case of human beings' height, we can see a range of that particular trait. We can't say tall and dark. We can't categorize into two. Instead of that, we can see a wide range of character. So the polygenic inheritance pattern is complex. It's very complex and it is difficult to predict the phenotype. We can't predict the phenotype. We can't say that the particular phenotype of a genotypic expression because there is a wide range. And the statistical analysis can give the estimate of population parameters. Okay, the statistical analysis that can give estimate of population parameters. So polygene means multiple gene, they have cumulative effect, and from that all these multiple genes. Don't get confused. I'm not talking about multiple alleles. I'm talking about multiple genes. So many genes are involved. Okay, not alleles. So multiple genes, this will have, each gene is having their own contribution in the final expression. And there is no epistasis. That means there is no masking effect, no dominance, and no linkage. But there will be contributing in each gene because that is a final expression of different genes. So in different genes, they have different alleles. So there will be contributing alleles of each gene and non-contributing alleles of each gene. So these contributing alleles, they are called as active, and non-contributing, we will call it as null alleles. Okay. And there will be continuous variation because multiple genes are involved and these genes will be having different alleles with variation. What do you mean by alleles? Different forms of same gene, right? So here different genes are there and different genes will be having different alleles. So there will be continuous variation in the phenotypic expression. Okay. And the polygenic inheritance pattern, that pattern is also complex because there is continuous variation. Due to that, we can't predict the pattern or the phenotype. Okay. And the statistical analysis that can give estimate of population parameters. So these are all the characteristics of polygenic inheritance. And next, we can see some examples. Examples of polygenic inheritance. Which is that example? Kernel color in wheat. So we are going to discuss about kernel color in wheat. In wheat kernel, that is a polygenic inheritance. Okay, polygenic inheritance. What is that? Quantitative inheritance or polygenic inheritance? An example is kernel color in wheat, kernel color. So here, the scientist which I said, Nelson, and one more scientist we told. Yeah. Among those scientists, Nelson, he is a Swedish geneticist, okay? And he crossed red kernel wheat variety. Red kernel. He crossed red kernel wheat variety with white kernel. Okay, red kernel with white kernel. And in F1, what result he got? In F1 generation, grains were red. Okay, in F1 they got red grains, but it was not like here we crossed the parental red. These are parents. Okay, here red kernel we crossed with white kernel. In F1 generation they got red color. One of the parental character that got expressed, but that was not exactly the parental red color. Okay, that was the difference. This was intermediate red color. Intermediate red color. Okay, that was the change or difference. So F1 generation grains were red, but it was intermediate. That means between red and white color of parental generation. Here again, that showed some blending. It was not exactly like the parental red color. Usually, according to Mendel's law or Mendel's experiment, exactly F1 shows exactly one parental character, right? But here, what happened in F1 that showed red color, that showed one parental character, but it was not exact copy of that parental character. Instead of that, that showed intermediate red color. So when members of F1s were self, this one, this is the symbol of selfing. Okay, when F1 members were self, F2, in F2 they got different ratio. F2 showed ratio of 1:4:6:4:1. Okay, this much only you need to learn. If you are writing the cross and explaining means it will be confusing for you people. So this ratio you need to learn. And red kernel when we cross with white kernel. Scientist Nelson when he crossed red kernel with white kernel, F1 showed red grains. It was red grain. It was red color only, the parental character, but it was not exactly the parental character. It was slightly different. That was an intermediate red. Okay. Intermediate between white and red. That combination. So, according to Mendel's law in F1 offspring should show one of the parental character. Exactly one parental character should be expressed. But here there is a slight change that showed red color, but it was intermediate red. Okay. When these F1s allowed for selfing, they showed a particular pattern. 1:4:6:4:1. 1:4:6:4:1. This was the pattern. So in this ratio, you can see out of that 16, we used to draw the Punnett square, right? With 16 columns. In that, 1/16. 1/16, extreme red. Extreme red. Okay? Means exactly like parent. Okay. Then the next one, 4. 4/16. 4/16, deep red. deep red. That means very dark red. Very dark. It was very dark. Here, parent, we have normal red and normal white, right? One showed extreme red color, like exactly like parental red color. And next 4, out of that 16, 4 showed deep red color, which is more than the parental red color. So it was very dark. Next, here next ratio 6. 6 out of 60. This represented intermediate red. Okay. Intermediate red, like F1. Okay, F1 we said it is intermediate red. So it was like intermediate red F1. Next one is 4. 4 out of 60 light red. That showed light red color. Then last one, this one. 1 out of 60. This is like parental white. Parental white color. Okay. So how many, in which ratio you got? F2, 1 is to 4 is to 6 is to 4 is to 1. So in this ratio, 1 out of 60, that was extreme red. Means compared to the parental red, it was extreme red. Then deep red we got. Sorry, 1 out of 16 extreme red. Extreme red means this extreme red which we are explaining like our parental red color. Maximum like a parental red color. Exactly like parental red color we got. Next, 4 out of 60. They got deep red. This deep red means it's deeper than the parental red color. Okay, very, very dark. It was very, very dark. So out of 16, 4 showed deep red color. Compared to the parental red color, it was very deep and dark. Next, out of 16, another 6 showed intermediate red color. Intermediate red color means it is exactly like F1. In F1, we got intermediate red color. Then 4 out of 16, they showed light red color. Light red color, we didn't get expression here anywhere. Up to F1, we didn't get anywhere. And 1 out of 60. This, in all Punnett square, we used to get that recessive character, right? So like that, white kernel, like another parent, they got the white kernel. Okay. So this was the ratio which they obtained while during their cross between red kernel and white kernel of the wheat. Okay. Wheat kernel while crossing the red kernel wheat and white kernel wheat. During that experiment, they got the ratio F2 ratio 1 is to 4 is to 6 is to 4 is to 1. This ratio, this is the polygenic inheritance ratio in wheat kernel color. You should note down this ratio. You should remember this ratio. Based on this, you may get problems. Okay. Genetics problems you may get. And next, see, Nielsen did all these experiments. So he found that this kernel color in wheat that is determined by 2 gene pairs. Which are those genes? 2 gene pairs. They were governing the kernel color. That 2 genes are kernel color is governed by 2 genes. Which are the 2 genes? Gene A, and this is one gene and this is another gene. Okay. So the dominant allele A and B, these determines the red color. Here, see, these are the 2 genes and the dominant allele A and dominant allele B. These determines the red color. Okay. This determines the red color and it is dominant over recessive allele. Okay, the recessive alleles, which will be the recessive allele A and B. These capital A and capital B, these alleles are dominant over that recessive allele. So the presence of allele A, dominant allele A and dominant allele B, that determines the red color of wheat kernel. Okay, so each gene pair shows Mendelian segregation ratio in this. So heterozygote for two pair of genes, that is when it's in heterozygous condition. For two pair, which are the two pair? Here we said A and B. So when it is heterozygous, means this is the heterozygous condition for first gene and this is the heterozygous condition for the second gene. Then they will segregate in a ratio. The previous one we said about the homozygous condition. Okay. When it is in a homozygous condition. Here when it is in a heterozygous condition, that means in this, which ratio we will get if it is in a heterozygous condition? We will get 15:1. 15 red and 1 white. Okay, this is the ratio. When it is in a heterozygous condition, polygenic inheritance ratio 15:1. So this degree of redness, that was depending upon the number of dominant alleles. Okay, we got so many, like extreme, like parental red we got. Then parental red. Then we got another type, dark red. Then intermediate red. Then we got white, right? So the presence and absence or the degree of that red color, that is determining by the presence of these two alleles. Okay, these two alleles, the dominant alleles determines the red color. Okay. Then another example. Here another example for polygenic inheritance is skin color. Skin color in man. Okay. In human being, you will get different type of skin color. So skin color is determined by Mendelian. According to Mendelian ratio, how that we should have an idea before learning all this examples. We should have a normal Mendelian ratio in our mind. Okay. We should have an idea about the normal Mendelian segregation. Then only we can find out the difference between all these things. When you are getting a question, they will combine both this Mendelian concept and this new concept. Then you will get confused if you don't have a proper idea about the normal Mendelian ratio and this polygenic inheritance or the quantitative inheritance ratios. Okay. So the skin color is, you know that skin color, it is determined by which pigment? Melanin. Melanin pigment. Okay. This is present in skin. Melanin pigment is present in skin. So the skin color is determined by melanin pigment. So the amount of melanin in each individual, that is determined by 3 pairs. Okay. Amount of melanin. It is determined by 3 pairs of genes. So melanin, which determines the skin color, and the amount of melanin present in skin, that is determined by 3 pairs of genes. Okay. So all these 3 genes, they are present at 3 different loci. So we said these are not multiple alleles. Okay, multiple alleles means that alleles will be present on same locus because they are different alleles of one gene. But here we are talking about different genes, polygenes, polygenic inheritance or polygenic inheritance or quantitative inheritance. That means more than 2 genes are involved. And these genes, in the case of skin color in human being or in skin color in man, these 3 genes are present, which is controlling melanin pigment, And these 3 genes are present at 3 different loci, 3 different locus. Okay. These 3 genes, they are from 3 different loci. 3 different loci. Okay. So each gene is responsible for the synthesis of a fixed amount of melanin. Okay. Here we said 3 genes are there, which is responsible gene 1, 2, and 3. All these 3 are responsible for the synthesis of melanin. But each gene is responsible for the synthesis of a fixed amount of melanin. Gene 1 that will produce this much of melanin, and gene 2 this much, and gene 3 more than that. So all together there will be a total quantity or total amount of this melanin, right? Suppose this first gene didn't synthesis, or that didn't synthesize melanin, means there will be a less quantity. That quantity will be less from this, the total amount. So like that, each gene is having, see, this is the total produced melanin. All 3 genes are having 3 different rows. This is A, or this is 1, this is 2, and this is 3. This much is synthesized by third gene, this is from second gene, and this is from first gene. Like that. By chance, first gene is not synthesizing, there will be less quantity of melanin. Got it? What I'm saying? So each gene is responsible for the synthesis of fixed amount of melanin. Each gene is having fixed amount of melanin. Okay. So the effect of all this gene, that is again cumulative or additive, and the amount of melanin produced, it is proportional to the number of dominant genes. Okay. When these genes are coming in dominant condition, all together this will work and it will form a particular amount of melanin. Okay. So the effect of all these genes, that is a cumulative or additive effect. So it is proportional to the amount of melanin which is produced is proportional to the number of dominant genes. So the total amount or the total quantity of melanin produced, that is quantity of melanin, is proportional to number of dominant genes. Right? Melanin, which is the skin pigment, and the amount of melanin, it is controlled by 3 pairs of genes. And these 3 genes, all these 3 genes, when it is in dominant form, they have different roles, or they are from different locus. All these are from different locus, and their cumulative effect, that will work out for the total production of melanin. So each gene is having a particular role. That means each gene synthesizes a fixed amount of melanin. Okay. So the quantity, total quantity of melanin, it is proportional to number of this dominant alleles or dominant genes. Okay. Here you can see different colors and the skin color. Okay, different types of skin color you can see here. So here, if you are taking an example of a pair of alleles of 3 different unlinked loci, I said no, that is from different loci. So these different alleles, how we can: dominant allele capital A, recessive allele small a, dominant allele capital B, recessive allele small b, dominant allele capital C, and recessive allele small c. So these are the different alleles of these 3 different genes. And this capital letter that is representing the incompletely dominant allele for dark skin color. Okay, it won't form complete dominant allele. Incompletely dominant allele for dark skin color. And the more capital letters, where the number of more capital letters are forming, there, see, like here you can see 3 capital letters. Here 2 capital letters and 1 is small letter. That means here the last one is recessive allele. Here all 3 dominant alleles are present, right? So like that, more capital letters show skin color towards the darker range and small letters towards the lighter color. Melanin is a dark pigment, right? So if all 3 genes, dominant genes are present, that will produce more melanin. That means it will become dark. That skin color will be dark. Wherever the number of dominant alleles are less, there you can see more skin color. See here, all are recessive alleles. So melanin synthesis is very, very less. Here you can see only 1 dominant allele. Here melanin synthesized, but only this much in this shade. Okay. And the parents, they are having genotype A, B, B, C, C. That means all dominant, and here all recessive. That will produce offspring in intermediate color. Okay. In F1 generation, in heterozygous condition, this is the genotype. Capital A, small a, capital B, small b, capital C, small c. This is the F1 genotype. And in F2 generation, 2 triple heterozygotes. Here you can see the same heterozygote. F1 is selfing, right? So in F2, they will give variety of phenotypes. Here you can see variety of phenotypes with different skin color. You can see there's a wide range here. That's why we have given the graphical representation. So variety of phenotypes you can see, and these phenotypes, they vary from dark to very light in the ratio 1:6:15:20:15:6:1. So this is what I said. We can't tell particular distinct character in the case of polygenic inheritance. There is a wide range. Okay, that's why these, out of all these, 1 it's dark and 1 is very light. You can see here. And if all 3 genes are in dominant condition, all 3 alleles are in dominant condition, that will be dark, because melanin, it is a dark pigment. When it is synthesizing more, the skin will become automatically dark. And when 1 allele is less, 2 alleles are dominant, 1 allele is recessive, then melanin pigment synthesis will be less compared to the first one. All dominant condition will produce more. When the dominant alleles are less, like that, melanin synthesis also will get reduced. Okay. So the ratio is 1:6:15:20:15:6:1. This is the skin color in man in this ratio, very dark to very light range. In this range, we got the skin color when we crossed. Okay, so this is the concept. Polygenic inheritance. So when we are taking a pair of alleles of 3 different and unlinked, these are from 3 different loci, but these 3 genes are controlling the melanin synthesis. Okay, and the capital letters, we are representing capital letters, capital A, capital B, capital C. Okay, when it is in a dominant condition, that will produce more melanin. This is the dark pigment. So wherever all these 3 dominant alleles are present, dark color will be more. And when like this, A, B, and 1, while crossing, that self-fertilization, when they are getting, if they are getting this combination, like 1 is recessive, then it will be having less production of melanin, so that the dark color will get reduced. Okay, it won't be as dark as the this combination, like all 3 dominant alleles. Got it? So parents having genotype all dominant and all recessive, that will produce offspring of intermediate condition, that will be having intermediate color in F1 generation. So in F1 generation it will be in heterozygous condition, and when F1 is getting self, in F2 generation 2 triple heterozygote mate. Okay, triple heterozygote. There are 3 genes and these are in heterozygous condition that will mate together, and this will give rise to varying phenotypes. Okay, you can see different color range. Here you can see light, again it's going towards dark side. So you can see different ranges of this skin color from very light to very dark, you can see. Okay, so that is in the ratio 1:6:50 1:20:15:6:1. This ratio, you have no other option. You have to read all these ratios and you have to learn the skin color ratio in man. You may get questions for your NEET exam and you will get combined questions of both concepts. So it should be clear. Both concepts should be clear for you. Okay. You want me to repeat anything? You have any doubt? Is it clear? Or should I repeat this polygenic inheritance? Deepa, is it clear? Okay, hope it's clear. If you have any doubt, you can ask me in the next class. The next class we will be learning about chromosomal theory of inheritance. So in that we will get an idea how these characters or genes are getting inherited or how it is going to next generation. So up to here we learned about the genes and how it is getting segregated. Next we are moving to chromosome level. So be clear up to that. You have to read properly. So next we are moving to chromosome level. So you should be clear with the gene concept and allele concept. If you have any doubt, you can ask me in the next class. We'll meet in the next class. Thank you.